In this lecture, we're going to look at synaptic connections. How do we characterize a synaptic connection? And briefly look at what stochastic synaptic transmission is and some caveats. So first of all, before we go into looking at synaptic connections, it's important to understand that the location of synapses, where they are, is extremely important for what the cell body will sense and hence react when it sends the input further. So the location is a very important feature, and it's not just enough to say neuron A is connected to neuron B. In the case of detailed morphology, the location is extremely important. And here what you see is experiments, very elegant experiments done by Stewart and Sackman, where they put an electrode into the cell body and then an electrode into the dendrites, and they evoked this action potential in the cell body, and it traveled backwards because we can see the delay, and then you see it in the dendrite after. So it basically travels back. It's called the back propagating action potential. This is very important because this is a signal that mediates plasticity and many, many other things, and it's a very important part of a lot of learning theory today. So this is a key experiment that is related to the current understanding of how neurons learn, but it also illustrates that when information, if this propagates back with a delay, it's going to hit the different synapses at different times. So synapses that are sitting at different places could be affected in very different ways when action potentials propagate back. And this is another example. If you had an input here in the dendrite and you inject a sharp current in the dendrite, then this is what you see in the cell body. And Edan Segev is going to give you all the theory and the mathematics behind what happens from here to here. But for now, what is important to understand is that if you had synapses here, it's quite different from having a synapse here. So location is important, and this rationale is important for how you characterize a connection. In the neocortex, there's actually only one connection out of thousands that could exist, that has been fully and extensively characterized. And even in our lab, we only did it once. And then we did it with less and less data subsequently. So this is this paper, which is a comprehensive characterization, and I'll use it to illustrate what would be a sort of a comprehensive characterization of a synaptic connection. So you record from two neurons. This is your one pipette, your second pipette, and then this is the axon going from one cell, and they have collaterals which you can't see here, and these collaterals move up and they form synapses on this other neuron. And when you excite this cell, you produce an action potential. Then in this cell, you see this response occurring. And every time there's an action potential, you can see sometimes it's a small response, sometimes it's a bigger response and a bigger response. And so this cell is sending a message to this cell, but it's receiving it with a different delay, a latency, which is here. It takes about 1.5 millisecond for the information to get across to the cell, but there's a distribution. And it also has different amplitudes. So it's stochastic in the sense that it has different amplitudes, different latencies, and we're going to explore why. You can do a much more higher resolution view where you have what we call the presynaptic spike, and then you can look at the response, and you can characterize exactly this delay across many different connections. Previously it was just in one connection you can see these delays, but now you can see across many connections, and you can see there's a lot of range. So some cells will take 1 millisecond to communicate with another cell, Other cells may take 6 milliseconds to communicate. The same types of cells. So you've got this big range of time delays between neurons. Some responses will be very short, and some responses will also rise very slowly. So they could go very fast up and down, or they could go slowly up and slowly down. The amplitude also varies a lot, up to 6, and actually we've even recorded up to about 12 millivolts for this kind of connection. But most of them are around the 1 millivolt range. So typically when one neuron connects to another neuron, it excites it, it's producing a 1 millivolt depolarization, what we call an excitatory postsynaptic potential. We can go a little bit further, and when you study these carefully, so you're triggering the spike here, and you look at the response, and then sometimes it fails, nothing happens. If you look at the amplitudes that you get, you either get nothing or you get a distribution. So you get a range of amplitudes that can be generated inside a single cell. But then you have many connections where most of them don't fail. So this is rare for this type of connection. But there are many others that fail, and some of them can have a very high failure rate. So they try to communicate, but nothing's getting through. Here you can see an example also of a very strong connection. It's about 4 to 5 millivolts. But even on each trial, there's a change in the amplitude, and this is the range of the amplitude. You can see. So when neurons communicate, they communicate with a jitter, a range from 1 millisecond to 6 milliseconds, and the amplitude can range anything from 1 to 12 millivolts. And we need to understand those mechanisms and how to recreate those mechanisms. Okay, so now when you look at when you stain these neurons, as we explained before, how you stain them, you load a dye into these cells. This is a photograph under a microscope when you've developed this dye like a photograph, so you can see the cells. You can follow the axon and the collaterals here. You can follow them, and you can identify where the axon is touching the dendrite of the other cell. And these areas are what we call putative synapses, because at a light microscope level, you can't identify whether it's a real synapse or not. You can just say that because the axon is passing by and touching a dendrite, and the axon is forming what's called a boutone, it's very likely that that's going to be a synapse. But this can be verified by looking at doing an electron microscopic analysis. And here you see the presynaptic side with vesicles. These are synaptic vesicles you see here. And this is the cleft. It's very difficult in experiments like this to get very high quality EM pictures. And this is the synapse. This is the postsynaptic side. So you can verify that what we call touches, physical touches, are actual synapses at the electron microscopic level. Now what is important in this is that when we did this analysis, what immediately became clear is that one neuron connects with another neuron by putting many synapses. So this is not a point contact, it's a hand contact. Lots of synapses onto a single neuron. And this is extremely important because it's a very strong rule in the neocortex. And you will see that this rule later is one of the most important rules that allows you to actually predict connectivity in the whole circuit. So this is what's called the multi-synapse rule. And we're going to discuss a lot more about that in the future. The number of connections varies between different pairs of cells. So some of them can have on average 3, 4, 5, 6, 7, 8, sometimes 10 connections. So this is the distribution that you see. The probability that one neuron will connect with another neuron is also quite low. It's around, P connection is around 0.1 or 10%. And we will discuss in future lectures what combining this knowledge with this knowledge means for connectivity. It has actually a lot of relevance for understanding how neurons connect with each other. So this is just a view. You can see that when multiple neurons connect, there's a cloud of synapses that are formed. So you can imagine when one neuron communicates with all these others, it's actually triggering lots and lots of little voltages in all the other neurons. So this is not going to review how synapses work. This you should read up in textbooks. But just to remind you, you have the axon coming in. It forms a bouton. Actually, it pretty much doesn't look like this, but it has a bouton. And the bouton has got mitochondria, which you also see here. They are powerhouses. They produce ATP so that this thing can run. It's below a micron. It's like half a micron typically. And when the action potential propagates down the axon, gets here, the voltage shocks the release of these vesicles. And the neurotransmitter diffuses to the other side, in this case glutamate in an excitatory connection, and it activates receptors, different kinds of receptors. Here you have AMPA, NMDA, kainate. And when these receptors are activated, they open and ions flow in. Sodium, potassium may flow out, calcium may flow in, and so on. But let's look a little bit at the synaptic dynamics. So, as we said, we have one neuron. It has, in this case, 6 synapses. And if you isolate, and this you can do in a model, you isolate each of these that are located at different places on the dendrite, and you look at this one individually and this one individually, you can see that they have very different behaviors, different amplitudes, different rise times, delay times, and what you see at the cell body is also quite different. So the location is very important, and one has to bear in mind that when a single neuron communicates with a cell, each of the fingers that are communicating is actually exerting a different effect on the target cell. Okay, let's briefly look at stochastic synaptic transmission. This is a very big field as well, and there's a lot of very interesting ways to analyze how synapses work. And you use methods called quantal analysis, where, for example, if only one vesicle or one synapse fired, you would see this sort of response, and that would be called the quantal size. If 2 fired synapses released, you would see a double amplitude. If 3, you'd see a triple, and so on. And in systems where these are very nice and clearly separated, you see these bumps. But in the central nervous system, in the neocortex, because the synapses are in different places in the dendrites, this becomes blurred, and you basically see a very blurry distribution. So it's very difficult to do what's called quantal analysis in central nervous system neurons. And it's important if you can do it, because you can actually identify how many vesicles, what's the amplitude, what we call the quantal size, how much impact does a single vesicle have or a single synapse have on the postsynaptic side. As an exercise, try to answer for yourself the following question. What is the probability What is the probability of successful transmission if you have 5 synapses in a connection, and each with a probability of 0.5, releasing when an action potential comes along? So you've got 5 synapses, each one has a probability of 50% chance of releasing. What is the chance that you will have a complete failure? Or what's the chance that there will always be a success? Also calculate what is the mean amplitude if each quantile size was just 1. So what would be the average if you have 5, with each one with a 0.5% probability, they each produce 1 if they worked, what would be the average? And calculate what is the coefficient of variation of the transmission. You need help? Look at this paper. Okay, let's look at some caveats. So the first caveat is that it's very difficult to obtain detailed characterization of synaptic connections. What I showed you is one connection that has been extensively characterized, and since then we have only partially characterized up to about 22 other connections, and there are hundreds that remain. And that's one of the reasons why you have to do reconstruction, because you can actually predict most of those others now, and that's what we're going to do during the course of this MOOC and illustrate how that's done. You also cannot verify easily that when an axon touches a dendrite, that it is a real synapse, and you can't always do electron microscopy. So you do have to have data that tells you what's your confidence that this is a synapse. And then the last caveat is that classical quantum analysis, where you can kind of peel out what's the effect of an individual synaptic vesicle or an individual synapse, and the probability that it's releasing, the size that it's releasing, and how the whole connection works, is quite difficult because of the uncertainty about the locations. The locations are distributed. The number of vesicles that are released may be 1 or 2 or 3, and then you may have many synapses. So these things make quantal analysis more difficult in central nervous system, but it's not impossible. There are statistical approaches around it. And to summarize what we learned in this lecture, is the most important thing to remember is all synaptic connections in the neocortex are multi-synapse connections. They're hand connections. They're not finger connections. Synaptic responses vary because every release site is probabilistic. So the vesicle can release probabilistically. The different synapses can behave probabilistically. The size of the vesicle can also vary slightly, not hugely, but slightly. And then the location of the synapses on the dendrites can vary a lot. The third piece of information that you learned here was that the number of synapses per connection depends on the type of connection. So if I showed you an example where there's just pyramidal cells and they may form between 3 and 8 connections. If you go to a different type of cell, it may be between 5 and 20 synapses. And so every pair actually has its own range of numbers of connections. And then lastly, because of stochasticity, in both the latency and the amplitude vary with the action potential. One has to bear in mind that there's really a very noisy signal that's been transmitted from one neuron to another. Okay, that's it for this lecture.